Have you ever wondered what makes us behave the way we do? What allows us to remember our experiences, or to learn new skills, or even to experience emotions such as love or anger? The answer is chemical communication within our brains. Now, in the Swidler Lab, we are interested in understanding how those behaviors can be traced back to that chemical communication, which occurs between cells in the brain using chemicals called neurotransmitters. Now, to understand which neurotransmitters are actually at play, we can profile the chemical makeup of the brain. Now, the way that I do this is to perform single cell mass spectrometry measurements of cells from animals such as fish or mice. Now, even though all of these cells do share chemicals with each other during that communication, there are many different types of cells, all of which create different types of chemicals. For example, one type of cell may create the chemical dopamine, which is commonly used to actually motivate behaviors. Now, another type of cell may create the chemical serotonin, which is commonly associated with a happy mood. Now, since these cells do create different types of chemicals, their chemical profiles will differ from cell to cell to cell. Now, if we were to take all of those cells and analyze them at once together, a lot of that chemical variety would actually be lost. This is because any of the rare or low abundance chemicals would just be drowned out by any of the high abundance chemicals. So this is similar to trying to hear your friend talk to you at a rock concert. Your friend's voice is just going to be drowned out by all of the sound from the music. Now, in my case, I'm interested in understanding the full chemical complexity of the brain, and so I want to hear from as many chemicals as I possibly can. So the way that I do this is to use single cell measurements. Now, I perform these single cell measurements on thousands of cells taken from the brain, which are then spread out onto a microscope slide and then stained with a blue dye. This way I can see them within a microscopy image, which I then use to find their locations within the main instrument called the mass spectrometer. Now, the mass spectrometer is what actually allows me to detect that wide chemical variety within those cells. So the way that it does this is by shooting a laser at individual cells, one cell at a time, to release the chemical contents of that cell. Those chemicals are then detected, and then I can take those chemicals and apply them back to the behaviors that were shown by the animal. Now, currently, I'm doing this process to understand the chemical differences behind different parenting behaviors shown by a stickleback fish, which is shown in the slide. Now, my hope with this work is that I can relate my animal findings back to human behavior to better understand our own brains. Thank you.